This is a podcast from the Nuffield Department of Medicine. Dr. Arturo Reyes-Sandoval tells us about his research on a vaccine against Plasmodium Pvivax. Hello, Arturo.
Hello, Anna.
Can you tell us about the different types of malaria parasites?
Malaria is caused by the Plasmodium parasite, and there are around 200 species of this parasite, and they can infect a wide variety of animals, including birds, reptiles, rodents, and primates. Of these 200, only 4 are generally considered that can infect humans and cause malaria. These are known as Plasmodium Pvivax, Plasmodium falciparum, Plasmodium ovale, and Plasmodium malaria. Plasmodium Pvivax is the most widely distributed form of malaria in the world, and it can be present not only in Africa but also in the Western Pacific, importantly in highly populated areas of Latin America and Asia, where it's a big problem over there. Plasmodium falciparum is mainly present in Africa, and it's responsible for high levels of mortality, mainly in children. Malaria and ovale, these 2 species are less prevalent around the world.
And what challenges does Plasmodium Pvivax pose?
The main challenge of this form of malaria is called the hypnozoite. This is a very small structure that the parasite evolves into, and it uses it to hide inside the liver of an infected person. And the parasite can remain there dormant for days, weeks, months, or even years. And the fact that the parasite can hide inside a person without being recognized makes it extremely difficult to eliminate from the places where it is present. To give you an example, there have been countries where malaria has been tried to be eliminated, but and in these places both falciparum and Pvivax coexist. And when after different attempts for elimination, only Plasmodium falciparum could be eliminated, but Pvivax remained there, and this is because it forms these kind of structures, whereas falciparum does not do that.
And how far away are we from a vaccine?
This is very difficult to predict. Pvivax vaccine development is in early stages of development in comparison to Plasmodium falciparum, which is more advanced. For falciparum, for instance, in the 1980s, it was considered that by 1990 there would be a vaccine already, and it's been more than 30 years, and so far there is no vaccine available for falciparum malaria or for any other parasitic disease. So it's quite difficult to know. In my case, I am planning to develop a vaccine and test it in humans within 5 years, and if everything goes well and we succeed, we will be able to test it in field trials within 10 years.
And can your research help to develop other vaccines?
Absolutely. The kind of tools that we have available work in a different way compared to traditional vaccines. Traditional vaccines can stimulate mainly or only one arm of the immune responses, the adaptive immune responses, which are called antibodies. Whereas viral vectors can induce not only antibodies but also T-cell responses or cytotoxic lymphocytes. This type of cells can eliminate other infected cells when a parasite goes or a virus goes inside one cell. So for certain diseases, this kind of T-cell inducing vaccines could be very important to be able to prevent them. For instance, Chagas disease, which is a neglected tropical disease that is caused by another parasite, Trypanosoma cruzi. It can be highly benefited by a T-cell inducing vaccine. Another example I'm thinking of is dengue as well.
And what are the most important lines of research that have developed over the past 5 or 10 years?
In my opinion, our ability to produce genetically modified microorganisms. One example is, for instance, the recombinant viral vectors that we use as vaccines. at the Jenner Institute and mainly the Vector Core facility has been able to produce nearly 400 recombinant viruses in the last 3 to 4 years, and all of them can be used as vaccines. Another field of great interest to me is our ability to create recombinant parasites, and one of my favorites is a malaria parasite that can express a transgene or a protein from the firefly, and it can produce light, and we can follow up the infection in real time.
So why does your line of research matter? Why should we put money into it?
Because Pvivax malaria is a human tragedy. We are talking of a disease that is responsible for nearly 130 to 350 million clinical cases every year, and nearly 40% of the human population is exposed to this disease. It mainly affects children or young people in working age, so this disease leads to poverty. We are now very fortunate to have tools that can induce extraordinary immune responses that we had never been able to achieve, and I think it would be desirable to use these tools to eradicate eliminate this disease.
And how does your research fit into translational medicine within the department?
Well, the Nuffield Department of Medicine has a great tradition in vaccinology. It is hosting groups that are making a major progress in the development of a vaccine against HIV, malaria, tuberculosis, influenza, just to name a few. And I believe that vaccine development is one of the best examples of translational medicine, because we as vaccinologists are aiming at producing or developing one product that can be used in animals or humans to prevent diseases.
Thank you, Arturo.
Yes.